Today's short video will be about Mendel and Punnett and the work that Mendel in particular did with peas. One of the places we will start out with is Pisum. Genus Pisum has 7 pair of chromosomes. So I'm drawing a pair right here. And what we're going to point out is that for particular traits, like, for example, a round pea or a wrinkled pea, there will be a particular spot along the chromosome where that gene is coded for. And we'll refer to a particular spot as a locus. But let's take a look right here, and let's just suppose that this is the area that codes for round seeds or wrinkled seeds. The male plant or the male parts with the anther, you're going to have 2 possible variants or alleles, and I'll write that. 2 possible variants for that gene, and we are going to look more at what those variants are in peas, but right now, just looking at 2 of these alleles, when the pollen is formed, in the case of peas, or essentially when the sperm are formed at the end of a process called meiosis, each one of those individual male parts is going to get one allele or the other allele, but not both. So this is why, since we have a 50% chance of getting any particular allele, this is why we spent time flipping coins, flipping bottle tops to look at probability. We're going to take a look at what Mendel did in his first cross and how a Punnett square can help us figure out the probabilities of certain types of offspring. Now, in this day and age, we are privy to some information that Mendel wasn't. For right now, in terms of simplicity in explaining this, we're going to jump into 2 concepts that are very important. They are the concepts of genotype and phenotype. Phenotype is once there is a combination of genes, what is expressed? What does the organism look like? The phenotype that we'll be looking at here, and one of the first phenotypes Mendel chose to look at, was the shape of seeds. And he found that seeds generally were going to be round, these little peas you can pull out of the pod, or some of those were dented or wrinkled. Now, we know at this point that there can be 3 different ways that a round pea can be coded for, and that's where we're going to need these things called genotypes. So one genotype for a round pea would have 2 dominant genes. And when I talk about 2 things that are the same, the prefix homo applies. And so here we're talking about a homozygous dominant genotype. Homozygous dominant. So another concept we're introducing here is the concept of dominance. We could receive an allele that is dominant and an allele that is recessive. We could receive, we being the offspring of a pea cross, could receive a recessive and a dominant gene, one time from the male part and the female part, and another time in the opposite way. And finally, there is one genotype that codes for wrinkled peas, and those would be called homozygous recessive. Recessive phenotypes are expressed only when there is no dominant allele present. So, we're going to use this concept next to look at Mendel's crosses and how some of these results occurred. So Mendel had spent time seeing that he could get pea plants that bred true. So when they self-pollinated, he had a group of peas that produced round peas, round seeds. He had another variety that very consistently produced only wrinkled seeds. He took these two varieties and crossed them. So, as we discussed in class, row 1, removing the anthers, going to row 2, taking the pollen from row 2 and fertilizing plants in row 1. His first cross, we, in hindsight, know he bred two homozygous pea plants. So what we're going to represent over here is the possibilities of the ovules or eggs and the allele they would get. And we're going to express above these boxes, because this is the way Punnett squares work, the only way to get consistently true breeding wrinkled seeds is to have the homozygous recessive. You'll notice I use capitals to represent alleles that are dominant. I use lowercase letters of the same letter to indicate recessive. Now, here are the possibilities. With this cross, in a new zygote, in a fertilized egg, we have these possibilities. Now, this could be done by showing different types of sperms and different types of eggs. But we keep our alleles nice and neat here. To know where these letters come from, I'll just do a quick show. So on that first cross, your possibilities all come out to be looking the same, or what we call heterozygous. According to these Punnett squares, there is a 100% chance of getting a genotype with a dominant gene and a recessive gene. 100% chance. So a heterozygous offspring. At this point, we are going to go back to class and see what Mendel's results were in his first cross, and then also to take a look at what happens when he takes these offspring, the offspring of F1, and does a cross between these. So the results of Mendel's first cross was to come up with homozygous genotypes, and these all led to the trait or the phenotype of round peas. So now what we're going to come back and do is take a look at what he did with this first generation. He then did a similar experiment as number 1, removing stamens from one group of plants and pollinating so he could control things. But the genotypes that we know we have in this cross of the first generation are going to look like this. A dominant gene and a recessive gene coming, let's say, from the female egg, and one dominant and one recessive gene coming from the pollen. And if we take a look at what our probabilities are here, you will see that out of the 100% or 4/4, that 1/4 of them are homozygous dominant, 2 dominant genes. 1/2, 2 of the 4, are heterozygous. They have 1 dominant gene and 1 recessive gene. And 1/4 of the time, 2 recessive genes. And if we were to look at what we now know about phenotypes, 3 out of the 4 should come out as round peas, and 1 of 4 as wrinkled peas. In class discussion, we'll discuss the ramifications of this further. Thank you so much.